Hi, my name is Dennis Province, and I'm the Director of Education and Outreach from the IDEA National Resource for Quantitative Proteomics. And I wanted to just take a couple of minutes of your time to talk to you about the undergraduate summer internship in proteomics that we hold here at or on the campus of UAMS, right here in Little Rock, Arkansas. This picture that I'm showing you right here shows you basically four different buildings here. You've got the main hospital over here, and then you have the Walker Tower and the Cancer Institute right here. And then you have over here, you have the Biomedical Research Center, or called Biomed 1. These three buildings here compose a lot of biomedical research that is happening right here in Little Rock, Arkansas on the campus of UAMS. And I want to tell you about the internship that would allow you to participate and observe a lot of that research that's going on right here in Arkansas. First of all, what is the IDEA National Resource for Quantitative Proteomics? It is an NIH or National Institute of Health funded laboratory that's located right here on the campus of UAMS. And its mandate is to provide global proteomics to investigators. That is protein mass spectrometry. I'm just going to give you a brief introduction of what protein mass spectrometry is and proteomics here in the next couple of minutes, as well as tell you about this internship. And the reason why I'm doing this is because most people have never even heard of the term proteomics. And so how would you know that you want to apply for an internship doing proteomics if you don't even know what that is? So what we do here in global proteomics is that people send us biological samples. That can be tissue or serum or plasma, those sorts of things. And through a sample prep workflow, we can separate out the proteins, digest those proteins into peptides, and then separate those peptides by liquid chromatography and analyze them by mass spectrometry. Woo! That's a whole lot of work that I just said in one sentence. And then it doesn't even finish there. After we get that information from the mass spectrometer, we do a database search to find the biological information that is contained in these biological samples that we started with. The biological information is what proteins were found in those samples and how much protein. Well, what is proteomics and where does it fit in with the rest of science? Most of you have probably heard of genomics, the study of the genome or genetics. That is interested in information at the DNA level. There is also something called transcriptomics, which is information at the RNA level. And then the proteomics is interested in studying the proteome, which is really the study of proteins. And of course, underneath there, you even have the study of the metabolome, which is metabolites, like things like sugars and nucleotides, etc., etc. But we want to focus here on proteomics. So proteomics is the study of proteins. Global proteomics then looks at the identity and the abundance of thousands of proteins all at the same time. These proteins perform all the work in the cell: metabolism, transcription, translation, repair, etc. All the stuff that's actually being done in the cell, for the most part, is all done by proteins. And so they're very, very important. The cell function or phenotype is controlled by the proteins. And so this is what researchers want to study. Well, what type of researchers are interested in global proteomics? And so I just say here, biomedical researchers like cancer biologists. This is why the national resource is located in the Cancer Center. Clinicians, those would be doctors and those that specialize in working with patients that have diseases and different maladies. Or researchers that are interested in how the proteome changes during some sort of environmental change, or after adding a treatment, or due to genetic modification. So something as simple as changing the temperature. So this graphic that I'm showing you here is from a publication called Molecular Cell, and the title of this publication is Proteome Plasticity in Response to Persistent Environmental Change. And it shows that there are certain proteins that change the their shape, and then where they hang out and their function, all based upon whether it's high temperature or low temperature. That is pretty amazing. And these are the sorts of things that can be studied by global proteomics. Now imagine if you have somebody that has cancerous cells versus normal cells. You can also study those changes as well. Or someone that suffers from diabetes and you want to know how their cells function differently because of this disease. I want to show you for a minute here just what the output really looks like in proteomics. So one of the things that people look for is they want to know um, what am I actually measuring? So this is called a volcano plot. In this particular study, frozen kidney tissue was taken, and from that there was an elution of immune complexes that was then analyzed by mass spectrometry. Each one of these dots represents different proteins that were analyzed from these kidney cells. Now, some of these kidney tissues were healthy, and some of these kidney tissues were not. They had some sort of disease. There was some sort of protein that was blocking up the kidney. And so this volcano plot shows you each one of these dots represents a different protein. Down in these two bottom quadrants here is where all of the normal, non-really changing proteins hang out. It's up here in these places where you have proteins that are significantly abundantly different from the control versus the treatment, or the control versus the disease. And so in this particular study, tubulin and ammonium proteins were found to be culprits because they were the most significantly upregulated in this. And so they were the ones that the researchers then targeted for treatment. So that's just a brief snapshot of how you would use global proteomics to be able to answer a biological question. So what's the internship like? You'd spend 10 weeks with us on campus working in our laboratories alongside other scientists. You'd get your own research project that you'd work on for that 10 weeks. You would also go to these weekly luncheons where other undergraduate researchers are going to be present from across the campus. There's 20 to 30 of them that all work on campus during the summer. You would also be able to present your data by a poster or a manuscript or a presentation like one of our previous interns, Dana Ohm, is doing right here. Who should apply? Well, we're primarily looking for undergrads from Arkansas or currently enrolled in an Arkansas university. So if you're from Arkansas but you go to school someplace else, or you go to school in Arkansas but you're from someplace else, you could apply. Students interested in exploring a career in biomedical research and you're currently pursuing a degree in biology, biochemistry, chemistry, or some sort of related field that has allowed you to take a lot of biological science classes that would be a good fundamental background for you for this internship. We're also looking for students going into their third or fourth year of college or their fifth year. I'm not making any judgments here. In the fall of 2024, you need to be motivated, curious, and willing and eager to learn new things. The types of skills that you're going to be learning is, number one, you're going to be working on a team. You're going to be working with a partner. You're going to be working with us. You're going to be working with biologists, with chemists, with biochemists, people that are experts at instrument maintenance and making sure that the data is collected properly, as well as people that are doing the computer coding and the big data science on the bioinformatics end. You're going to be learning problem solving, as well as biological sample preparation, and then collecting data from these state-of-the-art million-dollar mass spectrometer systems. To put it in a nutshell, proteomics can really be separated into three different areas. You've got the sample preparation on the front end, where we take the biological sample and we turn that into peptides. And then from the peptides, we do the analysis on a mass spectrometer. Once that becomes data, then we can do our data analysis through our bioinformatics using our workstations and different programs that we have here. And you're going to learn all of that if you apply and come to this internship. What's the application process like? It is an online application. It requires filling that out as well as 2 letters of recommendation and an official transcript from the current university which you attend. Just want to shout out to some of our previous internship participants. So here's Humphrey and Audrey that were here during COVID in 2021. And then here is, again, Dana and Walker in 2022. And then this is Natalie and Thomas from 2023. And we're hoping your picture will be up here for the 2024 summer. Starts May 20th, goes through July 6th, but 26th, and more importantly, the application is due March 1st. So this is the website to go to apply. You could click that QR code. It will take you right to the opportunities webpage, or you could contact me if you have more questions. Dennis Province at proteomics_internship@uams.edu. Thank you for your time.